Have you ever wondered what the actual instruction manual for life looks like? I mean, the code that makes everything tick. Well, today, we're going to unlock it. We're diving deep into how it all works.
So let's start with the big one, right? What is the instruction manual for life? And how on earth does it work? I mean, think about it. What's the blueprint that makes you, you? How can one tiny molecule hold all that information and actually do something with it?
You know, for the longest time, what our genetic material even was, well, it was a total mystery. Scientists knew something was being passed down from parents to kids, but what was it? This question, this single question, kicked off one of the greatest detective stories in the history of science.
So the search for this secret took nearly a century, but you can see the clues piling up. It really started back in the 1860s with Mendel and his factors of inheritance. Then, fast forward to 1928, Griffith notices this weird transforming principle in bacteria. The plot thickens in 1944 when Avery and his team finally point the finger right at DNA. But the real smoking gun, the absolute proof, came from Hershey and Chase in '52, and that set the stage perfectly for Watson and Crick to drop their bombshell in 1953, the double helix.
And just like that, there it was. DNA, the blueprint of life. But here's the really cool part. The genius of DNA isn't just what it stores, it's how it stores it. Its structure is the key to everything. It's this absolutely perfect marriage of form and function. Okay, so to really get how this all works, you've got to meet the two main characters in our story, DNA and RNA. Think of DNA as the master blueprint, right? It's the priceless original, locked away in a safe for protection. Then you have RNA. RNA is like a photocopy of just one page of that blueprint. It's a temporary, working copy that you can take out to the construction site. And you can see right away, they're built differently, and that's totally on purpose for their different jobs.
Let's zoom in on that DNA structure, because this is where the magic happens. It's basically a twisted ladder. And the rungs of that ladder, they're made of specific pairs. Adenine, or A, always pairs with thymine, T. In guanine, G, always pairs with cytosine, C. A with T, G with C. That's the rule. And this super strict pairing is the whole secret. It's how DNA can make perfect copies of itself over and over again.
So after figuring out this incredible structure, one of its discoverers, Francis Crick, boiled down the entire process into this one beautiful sentence. The genetic information flows from DNA to RNA to protein. That right there, that's it. That's the heart of it all. It's so fundamental, it's known as the central dogma of biology.
So this central dogma gives us a super clear three-step flow of information. First step is replication. That's when the entire DNA library gets copied, like before a cell divides. Step 2 is transcription. This is where you make a copy of just one gene, one recipe, and turn it into that temporary RNA message. And the final step, translation, is where that RNA message is read and used to build an actual protein. So let's zoom in on those last two steps, transcription and translation. All right, let's dive into that first part of using the code, transcription. The big question here is, how do you get a usable message from that master DNA blueprint without messing up the original?
Simple. You make a working copy. That's all transcription is. It's the process of creating a temporary messenger RNA, or mRNA for short, from a specific DNA gene. It's exactly like photocopying one page out of a giant priceless encyclopedia that you're not allowed to take out of the library. Now, in more complex cells, like ours, there's actually a cool little editing step. The first RNA copy that's made is kind of like a rough draft. It has these extra non-coding bits called introns. So the cell does something called splicing, where it literally snips out all the junk and stitches the important parts, the exons, back together. What you're left with is a clean, final message ready to go.
Okay, so we've got our clean, edited RNA message. Now for the grand finale. How does the cell actually read this message and turn it into a protein that does something? This is the last and honestly most incredible step, translation.
To do this, the cell uses something called the genetic code. You can think of it as a universal dictionary. It's the set of rules for translating from the language of nucleic acids, which only has 4 letters, A, U, C, and G, into the totally different language of proteins, which is built from 20 different building blocks called amino acids. And this code has some truly amazing features. For one, it's read in 3-letter words called codons. It's also degenerate, which is a fancy way of saying it's redundant.
Multiple codons can mean the same amino acid, which acts as a great safety net against mistakes. But the most mind-blowing part? It's pretty much universal. The same code that works in a tiny bacterium also works in you. And it even has punctuation, a start signal to begin and stop signals to end.
So, translation is the process of actually building the thing. It's where the sequence of codons on that messenger RNA is read one by one, and a chain of amino acids gets built in that exact order. It's like following a recipe to the letter to bake a cake.
Now, this whole construction project is a team effort. You've got 3 key players. First, the mRNA. That's the instruction sheet, the recipe. Then you have the ribosome. That's the factory, the workbench where all the construction happens. And finally, you have the real hero, the tRNA. This is the master translator. It reads the mRNA's instructions and fetches the exact right amino acid needed for the assembly line.
So, when you step back and look at the whole thing, you realize that understanding this flow, from DNA to RNA to protein, it's not just some abstract biology puzzle. It's given us the incredible power to read and even use the code of life itself. And once we understood the language, what did we do? Well, we set out to read the entire book. This knowledge led directly to one of the most ambitious scientific projects ever, the Human Genome Project, a massive, global effort to read the complete human instruction manual from start to finish. And the scale of this thing is just mind-boggling. The project found that the human genome has over 3 billion DNA base pairs. Let me put that in perspective for you. If you were to type out that entire sequence, you would fill thousands of books. It's an absolutely staggering amount of information packed into every single one of your cells.
But here's the thing. You don't always have to read the entire library to find the information you need. That's where techniques like DNA fingerprinting come in. It's a clever shortcut. See, 99.9% of our DNA is identical to everyone else's. So this technique just focuses on those tiny, specific bits that are highly variable, the parts that make each of us genetically unique.
And this has completely changed the game in so many fields. In forensics, it can place a suspect at a crime scene. For families, it can solve paternity disputes with certainty. And for scientists, it's a powerful tool for tracing our evolutionary history and understanding how populations have moved and changed over time.
So when you think about it, we've gone from searching for some mysterious factor of inheritance to being able to read our entire genetic blueprint. It's been an incredible journey, and it leaves us with the most exciting question of all. Now that we can finally read the code of life, what's next?